And actually, while I have you, I'm going to probably tee up this next question to you, and then we can certainly work through our panel. Um, while both manual and automated methods can, achieve comparable cell viability and recovery rates, automated systems offer significant advantages in consistency, whether that be reduced contamination risk, improved process closure, what have you. So I want to talk a little bit about, the processing, the processing of thawed cells to deliver the patient dose from a manual versus automated QC sampling, process closure perspective. So my question for you is, how do automated thawing and processing systems compare to manual methods in terms of that cell viability, recovery rates, across the different cell therapy products?
Right. So I may give you a little bit of an indirect answer to your question, Aaron, and then maybe Pratik and Kate can add better answers to this. But really, the first part of my answer is that, as you and everybody else knows, automated systems come with a higher cost, right? Both capital cost and development costs. So I think that we need to keep that into consideration as we have these biotech startup companies that start in a preclinical space, and then we have to go through the early clinical space, and eventually we get to a point, I think, and Kate and Pratik can disagree with me, where we are ready to make an investment, right, in an automated system. And as much as there is a theoretical potential of iPSC-derived therapies to achieve scales that are large enough to justify the investment in an automated system, at least Century hasn't gotten there yet, right? So, so we haven't gotten to the point where we have needed in these early phase clinical studies automation. And particularly automation, to your question, Aaron, post-thaw, right? So the strategy that we've taken is actually to completely minimize the cell manipulations that are required post-thaw in clinical settings, right? And where then we are starting to focus our ideas and investments around automation is actually on the fill finish part of the operation, where we think that need will arise first.
Of course, sure. And, especially from a fill finish perspective, we will be covering that, of course, later in our conversation. But before we do, Pratik, how about from you? What are, agree, disagree? What are your thoughts on, like we said, the processing of the thawed cells?
Manual versus automated. So maybe I can start there by the premise is, what exactly is the desire to introduce automation? So if you go back to that original, why are we aiming to automate a unit operation? And that's where I think sometimes on paper, people maybe overestimate what they're hoping for from an automated system. A well-constructed manual operation can certainly be matched by an automated system, but there is a higher cost, there's a higher capital expenditure that, as was discussed already, that goes into automating a system. But also, there's usually more flexibility on the manual side than there's on the automated side. You have to build a recipe and you have to execute it. So if you want to modify your QC sample points, or you want to take a larger volume, that requires quite a bit of work on an automated system that might not require as much work on the manual side. So it really comes down to what you're aiming for. Usually it's really around process consistency, and it's also about maybe process closure that really determines if you can automate a process. And of course, that you've already mentioned, Aaron, there's a lot of benefit there if you think about the reduced contamination risk if you can close the process with an automated system. It also gives you flexibility in the environment, the GMP class clean room, if you will, on where you can operate these, do these unit operations. If it fully closed, then you have flexibility that maybe we can move it from one lab to another, as opposed to having a full grade A type area that you'd be doing the unit operations in. So it comes back down to what exactly are you aiming for? I guess I would say in caution not to run right towards automation just because it's automation, but rather really think about your end use and how you want to leverage it. What is it really for? And if it's around one of those things, then maybe it makes sense.
Yeah, sure.
And I would just add the comment there. I think that it's really good point that, you need to write time when you're integrating automation, and then think about how different systems you may be using that automate integrate with each other, because technology can evolve at different speeds. So depending on how you're manufacturing, how you're doing fill finish, how you're doing cryopreservation, at some point you need these systems to talk to each other, and they may or may not do that particularly well over time. So, once you spend the time and the effort to verify, qualify, or validate something, making sure that that's really what you want, where you want to be with your technology. So I think, that does also speak to sometimes a manual system is great until you really feel that you're ready to automate. You can definitely have operator to operator variability, and that can be a challenge. But I think the goal when you do a manual process is to get it such that you don't see that operator to operator variability, and you've locked down your manual system enough so that you can sort of avoid that. Because I think we all know with science, if you have one magic operator that can do something and no one else can, then your process really isn't going to scale very far. So, all things to sort of consider.